let's talk about the mechanism of transcriptional activation. And in learning about the mechanism of transcriptional activation, we're going to focus on CRP. Okay. So two questions to be addressed about regulatory factors such as the LAC repressor and CRP. How do they recognize their binding sites? Okay, and so the short answer to that question, as we'll see, is that they recognize their binding sites through helix turn, helix motifs, which bind the major groove. Okay. And the second question that we can address, and we'll address today, is how do they communicate with RNA polymerase? So once they bind to their recognition element, how do they send a message to RNA polymerase that it's time to transcribe or not to transcribe this promoter? Okay. And so the short answer is that they do so through direct protein-protein interactions between the regulatory factor and RNA polymerase. Okay. And so here we see the structure of CRP bound to the CRP site from the LAC operon. Okay. And so like most bacterial transcription factors, it actually binds to DNA as a dimer. So it dimerizes, so we see two identical subunits here that come together via a dimerization interface, and then each subunit then has a recognition helix, has a helix turn helix motif that binds in the major groove of the DNA in the two different halves of the recognition site. Okay. And what we can see is that the two halves of the recognition site are identical to one another. This is a symmetric recognition site. So this strand has the same sequence as this strand. Okay, another important point is that we see cyclic AMP bound here to each subunit. So that's the space-filling model of cyclic AMP. And CRP only binds to DNA when it's bound to cyclic AMP, because cyclic AMP actually induces a hinge motion between these two recognition helices, between these two so-called reading heads. And it's only when cyclic AMP is bound then that these two reading heads are spaced the correct distance apart to simultaneously bind to their two halves of the recognition element. Oops. We see these two regions here, there's a total of 4 regions, 2 on each subunit, highlighted in different colors. Okay, so in yellow here then we see AR1, so that means activating region 1. Okay, so it turns out that CRP contains 2 activating regions, activating region 1 and activating region 2, and we see one of those on each subunit. Okay, and these were defined by a genetic analysis. So if you just heavily mutagenize CRP and look for mutations that prevent activation without preventing binding to DNA, we call those mutations positive control mutations. They don't block DNA, but they nonetheless block the ability of CRP to activate transcription, and so we call those positive control mutations, or PC mutations. Okay. So mutations in activation region 1 then prevent CRP from activating transcription, but they don't interfere with binding to DNA. In other words, they fit the definition of a positive control mutation. So the question is then, what this question boils down to then is, what is AR1 doing? How is that region communicating with RNA polymerase? Okay. And so, a question that the experiment I'm about to present on the next several slides addresses is, could AR1 interact directly with RNA polymerase to stimulate transcription? Okay, and so this question was addressed by chemically probing the interaction between CRP and RNA polymerase, the so-called label transfer experiment.